And so chromatin. I'm going to share my screen so that way I can go to the slide that has chromatin on it. And so right now I'm just pulling up that PowerPoint. So this is slide 19 that's referring to your question. And down at the bottom is the chromatin that's located inside of the nucleus. And so chromatin is DNA. And that DNA, it comes in those two flavors. And the flavors are open chromatin. And technically we call this heterochromatin. And then euchromatin, which we call true chromatin, is the closed form. And you don't have to know the terminology, but basically when it's open, it's active and it's being able to be transcribed into protein. And so the active chromatin that's in this space as the light purple area, the nucleus is actively producing mRNA transcripts so that way it can produce protein. The other type of chromatin, the euchromatin, that's closed or condensed, are called chromosomes. And those ones, they're there to package the DNA so that way it can be passed off into daughter cells for mitosis or meiosis. And so that's essentially, chromatin as it relates to your test. And I'm also going to try to open up an exam so that way I can make sure that it is directly answering your question. But yeah, that's chromatin.
I wish there were more people to ask questions because I can't think of any at the moment.
You can use this time as a study hall, and as you're studying, if a question arises, then just feel free to just speak up and say. Um, I will tell you and the class directly that there are two questions that either have the terminology chromatin in the question or the answer. And so that's why I emphasize that slide with the nucleus.
Thank you.
Uh-huh.
Okay, I got one. I'm doing the mastering in my lab, and one of the questions says, which of the following processes and organelles account for the replacement of lipids and proteins lost from the plasma membrane? I kind of want to know, like, what is exocytosis?
So exocytosis is the movement of material from inside of the cell to outside of the cell. In the next unit, which is going to start for us on Monday, I'll explain this. And let me just make sure again that this is not covered. Awesome. So it is not on exam number 2, but it is on exam number 3, unit 3. And so, like I said, on Monday, we're going to cover it. But exocytosis, the way I'll explain it in class, is that your ex, like it could be an ex-boyfriend, a girlfriend, ex whatever, they leave. If you're on a plane and you sit on a seat above the wing or towards the doors, those are called exit rows, to leave. And so exocytosis means to leave the cell. And that's how the cell gets rid of things, either positive or negative. And so that's what exocytosis is.
Okay, thank you.
Mm-hmm.
Okay. The test goes over like concentration gradients, right? I just in class I kind of went one ear and out the other. Again, I have like a little bit more explanation on concentration gradients and like active passive transport.
Understood. Let me pull it up and then I'll share my screen. Okay. So this slide, slide 19, I wrote it on the board in the upper right-hand corner, I believe in red. And these are the three types of transport that you have to know. Um, Now, you already asked a question about uh, exocytosis. Technically, that falls under bulk transport, and uh, we haven't covered that yet, and so that's for the new material. But passive and active transport are covered in unit number 2 test, and then, like I said, we'll move forward. So passive transport, I think the best slide that describes it is this slide, and we call passive transport diffusion. And I wrote on the board that it does not require ATP. I sprayed the perfume in one half of the room, and then, without any energy, random collisions caused the perfume to be moved from an area of high concentration to an area of low concentration. And so, again, we call that diffusion. And the picture for this fishbowl and the little crystal, it it shows a great, like how it really happens, what's going on in the air as well. The other one is a facilitated transport. And this goes back to one of the slides from the previous part. It goes back to slides 17 and 18. And I also really, really emphasize slide number 17, and I had you write it on the board. And we're talking specifically about substances that cannot move through down here at the bottom. If a molecule is too large, charged, or polar, it can't move through, and so it needs a little bit of help. And that help comes from the proteins that are embedded in the membrane, carrier proteins and ion channel proteins, as well as, to a degree, the junction proteins. And so those three are going to help facilitate move large, charged, or polar things from a high concentration to a low concentration. It's facilitated diffusion, facilitated transport, the same thing. High to low, no energy. The last one is active transport, and I'm trying to think. Yeah, it is down here. Active transport, I just made an announcement about this because I checked and it is in the answer choices and or questions. And so I want you all to be prepared. Active transport moves against the gradient. This is where I said in class that the perfume was really expensive, $500. And I need to repay, I think it was Alani, the $500 by collecting all of those molecules and putting it back in. That takes a lot of energy. It takes a lot of time. And that's what active transport is. It takes energy in the form of ATP, and you have to move those molecules back to the highly concentrated bottle or to the highly concentrated area. And so that's active transport. The most important thing for you all is osmosis. And so this is the slide that I used, and it has the animation on the left. Osmosis is relative, and so you have to describe it in terms of the cell or the solution, and you can also describe it two ways, describing the water or the solute. And so in the bottom part, over here. Let me see. Go. So for this bottom part right here, we can describe this as H2O, or we can describe it as solute concentration. On the left, there's a low solute concentration, and on the right, there is a high solute concentration. On the left, there is a high water concentration, and on the left, there is a low water concentration. But the movement is still going from left to right. So it doesn't matter the concentration of either the water or the solute um, in terms of the flow. In this case, it's going from left to right. So water is moving from high concentration to low concentration always for osmosis. For osmosis, um, the solute concentration in osmosis is moving from low concentration of water to the high concentration of solute. And so that's two ways of saying it. And I hope that's not too confusing.
So,
So, mhm.
so you're saying like it always goes from high to low, but that doesn't determine like it could also be going left or right, like
Yeah,
whichever one's okay.
exactly. It could be going left or right, but it depends on the amount of water in either location or the amount of solute in either location. It just
Okay.
happens to be on this type, the solute is on the right, and so the movement is from left to right. But if we switched it, it would be right to left.
Okay.
Yeah.
I think I got it.
Mhm. And I like the pictures that are underneath. And uh, so the pictures that are right here to describe the actuality of osmosis are the most helpful because it's showing where the water is and it's showing where the solute is. And so in the first part of the picture here, this part, um, those have an equal concentration of water and solute, and that's why the arrows are equal. And so there's two arrows going in and two arrows going out. So it makes it isotonic.
I have a
Well,
quick question before you proceed. I'm sorry.
yeah, yeah.
Because I know you mentioned that hypo means too low and hyper means like, the other way around.
Hmm.
So why in a hypotonic solution does it say that it bursts? Like, is I thought that was supposed to be too little water.
Um, it's too little solute in the solution. That's what it's referring to because it's in reference to the cell. And so it's a relative amount. And so for the middle picture, this solute is super high, and
Okay.
the solute in the water is super low. And that's why it's hypo, because the solution is low in solutes. And
Mhm.
so the water is going to the high solute concentration, and that was, it's going to leave it up there, but that is this part over here, and the solutes are over here. So the water is always moving to the area of high salt concentration.
So what solution is it? You say this picture right here, that one that you just showed, that's a hypotonic? Or is it a
Exactly. It is a hypotonic solution because the solution has a low amount of solutes.
okay. Okay.
If we flip it, if we flip it and then go to this other one, then if we look at the solutes on the outside, now the solutes are a lot. And here the solutes are a little bit. And so water is always moving to the salt or to the solute. And so water here is moving out. And so there's more solute in the solution, so it's hyper.
Okay, so like with the picture before, because the water was trying to move into the solute, that's what makes it hypotonic.
Exactly. So
Okay.
the cell here, the cell here is super salty. So water is moving to the salt, to the solute. Here, the solution is super salty, solute. And so the water is moving to the solute, to the solution in this case.
Okay. Thank you.
Yeah, yeah. There is a phrase that people may have heard is called water follows the salt. That's why we drink Gatorade, because our body is super salty and dehydrated. And so when we add Gatorade or just regular water, the water is going to go into our body. But if we are very well hydrated, then we're going to pee out clear liquid because we don't need all of the salts. And so the salts don't chase our urine, and so it ends up being clear.
Okay.
Yeah. I hope that makes sense.
Yes, thank you.
I have another question. So I was doing a practice problem and it said, what would be the sequence of the strand of RNA that is made from the DNA template? And it shows 3 first and then GAT, AT, CGAT. And I know that the T's get switched out with U's, but I thought it went 3 to or 5 to 3 all the time.
Mhm. So that is, you said it went three first, and then it gave you a sequence. And can you say the sequence slowly?
Yeah, it's going to be GATA, TC, G, A, T, and then 5.
So all strands, kind of like amino acids, they all have a 5' end and a 3' end, or a 3' end and a 5'. It has to have that. But I told you all, in order to read this and in order to write this, it has to be 5' to 3'. This strand, we know that it is DNA because it has T's instead of U's. And so if we were to transcribe this, we would need to put in U's for all the base pairs that contain A's. That's first off. And then wherever there's a G, we're going to put a C. T's get A's, C's get G's, and A's or T's get A's. Now, this is complementary. So wherever there's a 5, because we complemented it, we base paired, the other strand has to be the 3' on the right, and that makes this strand over here 5' on the left. Because there are U's right here, that lets us know that this is RNA as opposed to DNA. And so that would be an example of transcribing something.
What is this? Is this just trying to identify which is which? Or
What I explained is the entire concept of DNA and RNA, but it's also talking about transcription as well. And we'll get more into this at the Unit 4 test.
Thank you.
Yeah. Did I answer your question okay?
Yeah. I thought that if a 3, like the 3 being on the left side meant that the 3 below it would be on the left side, and then 5, because I was like, it always goes 5 to 3. So that's what kind of confused me on that. So thank you for clarifying.
Yeah. In fact, what I'll do is another example right here. But I'm gonna go to my backpack in my chair because I need to get a charger for my laptop. 1 second. Okay. So I'm going to do another example now, and it'll talk to the point that you made, Brittany. So you're used to receiving something in the 5' to 3' direction, and when you're given it something in the 3' to 5' direction, it doesn't mean that it's wrong. It just means that it's the other side of the strand. It's the complementary strand. And so all you have to do is just add a 3' and a 5', just like we did for the mRNA that's down below. Oh, gosh. We're going to do the complement. So if we have a G right here, we're going to put a C. Wherever we have an A, because it's DNA, we're going to put a T. Because we have a T, A, T, T, A, C, G, G, C, A, T, and T, A. Now what we have are two double strands, or it's a double-stranded DNA. And I already wrote over on the bottom that that's a piece of mRNA because it contains the uracil. And so you could be given the 5' to 3', no big deal. or you can be given the 3' to 5', no big deal. It just turns out that later on we'll discuss that the 3' to 5' in this case is called the template strand. And so if you have a template for something like an art drawing stencil, you're directly going from one to another. And so we would call this the template strand later on. And so, yeah, that is kind of the exercise in it. If you're given the 5' to 3', then make sure that your mRNA, this is important for, I think, one of your future tests, is to make sure that you're going to flip these ends in the nucleotides in the middle, so that way it ends up being the correct orientation.
Brittany, I love you, but we're going too far in advance. This is chapter, exam 4. That's crazy. But it is cool to see how everything kind of ties together.
Mm-hmm. And I'll make sure I'm looking in the background right now, just to make sure that if it is represented on the test, and I know it is, but just the level of knowledge that you will need.
And you said the top two, the 3 GAT and then the 5C UA, those are mRNA?
The top ones that I circled in that yellow color, those are DNA because they
Oh, I
contain.
didn't see the top one, sorry.
Oh yeah, it contains T thymine for DNA.
Okay. Yeah, the middle ones though, you said were mRNA, right?
The one on the bottom is the mRNA.
Okay.
So yeah, the one that's in blue is mRNA, which is it's this one. So this strand right here, this is mRNA, and the two top ones, those are DNA. This bottom one in brown, that's also mRNA, but what I did was I just switched it. Like I literally flipped it 180 degrees. But I will tell you, full transparency, Brittany, just like Aiden said, I just looked over the test. It is not represented on the exam, like in this fashion, but it is coming. Like, so if you even store one brain cell of it, it'll make it easier for later on.
Yeah, this stuff, for whatever reason, makes more sense to me than like the molecules themselves. So
Yeah, it just means that you're going to kill it for unit number 4. Because this is when we talk about mitosis and meiosis. And what's actually happening is the DNA is doing this stuff. And so you need to know about DNA, but this part, don't worry about it. Don't stress. So now you know you don't have to study it.
So if you were if you were to like take all the concepts from from this exam, like what would you say are like the best ones, or like the ones that have like the most kind of questions to where that's what we should focus studying on, and like just have like a little bit of knowledge of other things?
Yeah. So I told a student via email how I would prefer that you test your own knowledge and see your strengths and weaknesses. And so think about where we started, like for this unit, and we started with functional groups. And so I want you to start writing out your functional groups. That's a carboxylic acid. And where do we find carboxylic acids? Well, we find them on fatty acids. Well, this is now a fatty acid. How many fatty acids does it take to make a triglyceride? Well, it takes 3. What else do you need to make a triglyceride? You need a glycerol backbone. Glycerol backbones and fatty acids make a triglyceride. A triglyceride is nonpolar. Nonpolar means that it hates water. And so this is what's called mind mapping. And so you're linking together 2 different parts that I've taught you. Over here on the left, I've taught you about functional groups. How does the functional group relate to the biomolecule? If we took it a step further, the biomolecule fats or these fatty acids could be making a fatty acid tail that is a part of a phospholipid. And so the phospholipid ends up making a cell membrane, or just membranes in general. I could even go a step further on the right-hand side and say that fatty acids come in 2 varieties, or 3 really: saturated, unsaturated, and in the unsaturated ones are cis or trans. And so by mind mapping, you're pulling together all of the concepts. If you can do this and not miss anything, like in terms of, like, I didn't leave anything out between chapter number 4 and chapter number 7, then you're good. And I can even go on and say that certain items right here are going to go through the membrane, and then other items, they're going to get rejected. What things can go through? The things that can go through have to be small, uncharged, and nonpolar. That means fats like steroids, testosterone, estrogen, and progesterone, cholesterol, they can be found in the membrane or they can go through the membrane. If they get rejected, that means that they're large, charged, or polar, and so they get rejected and they need facilitated transport, which ends the whole entire concept of what we studied. That was, you know, I think that was slide 30 in the PowerPoint out of 33. And then the other part would be, how do we get things to move from a high concentration to a low concentration? If it's large charge and polar, it needs facilitated transport. If it's going from low to high, we call that active transport. And active transport needs to be facilitated by proteins. And so, boom, that literally is everything. So that's how I would like you to study. And you won't miss a single question on the test like that, or at least up to mid-level. And you do practice questions to get the critical thinking and to know how to answer questions in general.
Okay. Yeah, because I just remember you said that this test like was going to be like, relatively easier than the last test. And when I was in class, I just felt like there weren't a lot of moments where I went, aha, I get it now, So definitely a lot of my own studying that I need to focus on.
Uh-huh. I'm trying to see if there's a picture that I'd be willing to show y'all again. But I've literally told you all of the questions, like how the questions are. Like when I was saying this is important, I'm talking about this one thing. Knowing pictures. Yeah, I kid you not. Brittany, you recorded the lectures visually and with audio. If you play back every single lecture and you can do the study guide, if you can mind map, if you can do practice questions, I promise you, you will see every item that I've put on the board and told you that's important for you to study, other than what you mentioned about like the transcription and translation. Throw that out the door and you'll be good. Mastering in my lab will give you another great direction. And I promise 85% of everything that I said, if you know it, you'll get it right. And the other 15%, maybe these really, challenging critical thinking questions where you'll take a minute to digest it and then hopefully select the right answer. Yeah, there's pictures and yeah, it's there. You have all the tools. You definitely have all the tools to do well.
Can you tell me, because when I was recording, I actually turned off before I could write down when you had like, I think it was page 17, 18, 30.
Mm-hmm. I can send you something.
Thank you.
Yeah,
Did you,
yeah.
did you say you kind of wanted to know like the slides he listed?
Yeah, and I can forward it to you, Aiden, if you need.
Yeah, these don't I didn't put like the exact chapter they're on, so you're gonna have to like, bear with me. So I'll say like from the start to the end, and so it'll probably be all on different chapters, but I see slide 13, I see 28 and 13. Those are probably for a different one. Slide 32, slide 33, slide 37.
Mm-hmm.
What
I'll
else?
send you all these because it's about to end. But
Okay.
I'll post this and y'all just check your email and it'll be there for you.
Okay, wonderful.
All right. Y'all do well. If you
Thank
need
you
any
so
help,
much.
just email me.
Yeah. Thank you so much. We're the only ones that were here, so we better get the highest grade. I'm just saying.
Okay, cool. All
All right.
right. Bye.
All right.